All right, so we're going to talk about DNA replication and chromosomes for our intro to zoology class. This is probably a pretty simplified version of what you should at least understand about DNA. And it's obviously remarkable for it being the blueprints for just about every protein or modified protein in your body for the most part. It is, when we talk about our nucleus, we talk about how there's these different chromosomes, there's this DNA, and literally the amount of information in the nucleus would be equivalent to going over to our library and looking at all the volumes. So it's this incredible amount of information that is stored inside of your nucleus. Now, it wasn't always understood that the DNA was actually the blueprints for your cells. There was all sorts of speculations. Where was it? Was it, you know, they had to speculate whether it was even in the nucleus or whether it was proteins, you know, and so forth. So there was a lot of discussions and trying to figure this stuff out in the 19th century. And of course, it's still a very important topic, but a lot of the basics are now understood. DNA is, again, deoxyribonucleic acid. DNA is made up of these nucleotides, and there are only 4 nucleotides that make up DNA. There's also ribonucleic acid that includes a different nucleotide in place of thymine. So the nucleotides are made up of purines, which include adenine and guanine. And for simplification, we always just call them A and G. And pyrimidines, such as cytosine and thymine, and which, again, for simplified versions, we call them C and T. So these are the basis and the internal structure that you've probably seen lots and lots of DNA molecules, whether it be from previous classes or even in pop culture. They're the centerpiece of the spirally DNA that we're all so familiar with and seeing. There's also ribonucleic acid that changes or takes the place of thymine. And then if you remember adenosine triphosphate, ATP, 3 phosphates. Remember, adenine would actually be part of that nucleotide plus 3 phosphates. So that's also used as our fuel source for the different operations of our body, whether it be moving our muscles or enzyme reactions, all sorts of things. So this is what the 4 nucleotides look like. Again, you might not had a very strong chemistry background, so just a quick reminder, this is basically organic biochemistry kind of stuff. And things that you should be familiar with is that, like, for each of these corners, we have a carbon atom for shorthand. And then each one of these lines represents a covalent bond with 2 electrons. And so this corner right here would be a carbon atom that's covalently bonded to this carbon atom and that carbon atom. You should know this, and if not, you'll learn in chemistry. Carbon atoms can hold 4 bonds, 4 covalent bonds. So if you look at this carbon atom here, each one of these lines represents a covalent bond. And again, if you're not familiar with covalent bonds, you'll learn about it in chemistry, or just take a few minutes sometime and read up on it, because it's something you should at least have some basic familiarity with it. I hope that you learned it in high school. And so sugars are made up of these carbon atoms with OHs attached to them. So are alcohols, by the way. And so a carbohydrate is a hydroxyl group. So there's basically a 5-carbon sugar here. And it would be ribose. Remember, it's deoxyribonucleic acid. That's the ribose, the ribo. Look, there's a phosphate. Remember how adenosine triphosphate would have 3 phosphates? Well, in this case, it has a phosphate backbone. and then it has a nitrogenous base. And remember, there's purines and pyrimidines. Here we have adenine, thymine, guanine, and cytosine. And so the differences you'll see by looking at the nitrogenous base. And if you go back, do you remember which ones were the purines and which ones were the pyrimidines? Well, adenine and guanine were the purines, and cytosine and thymine are the pyrimidines. It says large base. So if you go back over here, you can see this is a purine. See the large base has two, well, keep it simple, two circles full of carbons and nitrogens and hydrogens. So this guanine is a purine. And then these are pyrimidines, adenine and thymine. See how there's only one. So anyway, those are the 4 nucleotides that make up DNA. And so it's kind of like having an alphabet, but it's based on 4 letters. And those 4 letters in all sorts of different combinations brings in amino acids. I think it was roughly 20-something amino acids. And they make up all the different types of proteins that are in your body or other organisms' bodies. And so looking back in time, they noticed some basic things as they started studying DNA, that it seemed like purines, well, it didn't seem, they noticed that purines and pyrimidines equaled the same number. There always appeared to be the same number of A's and T's and the same number of C's and G's. And so they called that Chargaff's rule. And eventually what we find out is that these nucleotides attach to each other and forming the DNA inside the center of the DNA alpha helix. Rosalind Franklin was very important in helping discover DNA. She did X-ray diffractions that revealed the DNA shape and that it had a helical shape. Unfortunately, she died, and probably in part due to her field with radiation and things like that causing cancer. But her image, and I don't think it was borrowed properly, gave Francis Crick and James Watson a lot of information on how to figure out what a double helix looked like. And so, because there was basically a race to figure out what DNA looked like and how it worked, of course. And so by using that picture and lots of other people's work, and science is about that, but how they went about it might not have been the most appropriate. Irrespectively, they were able to deduce that it was a double helix. There was all sorts of different ideas before. And they didn't really know. I mean, how would you go about trying to figure something out like that? It was rather complex, as you can imagine, requiring people's lifetime of research and study. So looking at all these different conclusions from Chargaff and Franklin and Tinkertoy models, using Tinkertoy models and making covalent bonds with it, they were eventually able to come up with what the shape must look like. And of course, this was a very big deal for them. And I believe James Watson's dead now. It's been since the last time I updated these slides. But anyway, they were able to, along with the work of others, and that's how science does work though, come up with the shape of what DNA would look like, the alpha helix or double helix. So this is a picture drawn, of course, of what a double helix looks like. Remember what these bases are? Obviously, the A means adenine. And is this a purine or a pyrimidine? Well, clearly it's a purine. And then over here, across from adenine is a thymine. So we see this relationship over and over again. And I'll explain what these dots here are what we call hydrogen bonds. So that means there's a slight attraction between these two molecules to each other. It's not as strong as a covalent bond, but it's kind of an electrical charge that helps hold them together. So adenines have this electrical charge that helps them to stick to thymine. And then guanine, and you'll see there's 3 hydrogen bonds, helps it to stick to cytosine. And you'll see this repeated over and over again. Now, again, it's a double helix because it has 2 sides to it. So it's almost like if you had 2 strings, 2 shoestrings that you wrapped around each other. And then they're sticking in the middle because of these letters. And then you can actually unzip it. You can actually unzip these letters and pull them apart because it's a weak bond. So it'd be kind of like Velcro. You know how you can pull Velcro apart? Hydrogen bonds kind of like Velcro, while the glue that the Velcro is attached to is a lot thicker and heavy, more like the covalent bonds. So you can unzip it and break these coils apart. And that happens when the DNA replicates inside the cell. There are obviously a lot more to it, but this backbone is made up of our deoxyribose sugar and phosphate atoms. And again, these lines would represent covalent bonds. And of course, there's carbon atoms here and hydrogen oxide, hydroxyl groups, and so forth coming off that you don't see. It's just made into shorthand. So there'd be all sorts of atoms making that up. And then as this gets wrapped around, and you have what you call minor grooves and major grooves and so forth. But the take-home message is the backbone is made up of the sugar phosphate group, and the thymine attaches to adenine, and guanine attaches to cytosine. The bigger base is the purine, and the smaller base is the pyrimidine. You'll also notice, and this is, you'll learn more about this when you take cell biology, but you have a 5' end and a 3' end. And so what that ultimately means is that they're facing in slightly different directions. When you look at the ribose sugar, notice how it's pointed this way. And then if you look at this side, it's pointed that way. So it is pointed in slightly different directions when they come together. So it'd be like my 2 fingers or 2 strings going like this, not like that. That makes sense? Because they're facing in different directions and then they stick together, all coiled up. That's what we're seeing in this picture. Now, mutations mean that sometimes you get different letters in here, different bases. But anyway, the X-ray diffraction helped to give them this information. Here's another picture of the double helix. It's not shown here, but what is in this backbone again? Phosphates and, hang on, mate. Go back to this. Sugars, phosphates. So we know there's phosphate sugars, and then this is our basis. These little dots represent the hydrogen bonds. And again, if you don't know what that is, take a little time to read up on that. Just Google it real quickly, or you'll be learning about it in chemistry. So again, the DNA molecule, and we're going to talk about replicates. But the key is to understand that there's these complementary base pairs where adenine always pairs with thymine and guanine always pairs with cytosine. And so if I gave you a strand of letters representing the nucleotides, ATA, CG, CAT, you should understand that without even me showing you the bottom, that A is going to have a T below it. And then T is going to have an A. Again, these nucleotides are just like letters as far as analogies go. And so these 3 letters might represent this particular amino acid needs to come in, and then this 3 letters represents another amino acid. We call those codons, by the way, because they code for a certain amino acid. Anyway, talking about the DNA molecules, remember this would have hydrogen bonds, and then the backbones and all that stuff would be back there. So it's basically kind of a mirror image, is what we call this. And so you can literally, the reason why you can do DNA technology and do things in a test tube is you can take a strand of DNA and make the other side with enzymes and nucleotides in a test tube up in my lab. We do it all the time. So either side can make a template for the other side. Does that make sense? So if I was just to remove the top and you had the bottom, you would know A goes here. T goes there. But they didn't really know how DNA replicated. They had all sorts of different ideas. They wanted to know, how does DNA replicate? When does DNA replicate, by the way? What were the two processes we learned? We know DNA is going to replicate before mitosis, right? And we know DNA is going to replicate before meiosis. So how did that happen? They had to figure this stuff out. So before we were ever even thought of, or at least you all, they were trying to figure that all out. I guess that's probably me too. But, and so they went through all sorts of different ways where they could use, I wouldn't call it painting, but they could make some DNA a little heavier than other DNA, radioisotopes and all sorts of stuff. And that would get an idea of what type of nucleotides were coming together and how they were showing up. So looking at this slide, there was all sorts of thoughts about it. Would the original DNA unzip itself and make two strands and then make a brand new, pure new strand? Because remember, follow the colors. Here we got blue. And then we make a new, and then we unzip it, take the Velcro and pull it apart, put the nucleotides in, the new nucleotides from the nucleus that be floating around, and then cut that piece off and stick it together and then put these pieces back together. So this would be the original and this would be the new. That was one idea that took place, but it was called, and that idea was called conservative replication. But I'm going to put a big X through it. That's not how it works. That was just a thought. They did lots of different experiments ultimately to figure that was not the way it happened. Again, the way I told you is you would have painted, in quotes, one side red and one side blue, or the original blue, and again, painting means giving maybe a radioactive heavier isotope or something like that, and doing all sorts of experiments and figure that out. There was also the semi-conservative replication idea and the dispersive. The dispersive is you would have the original in blue here, and then there'd be a little bit of red, a little bit of blue, and it'd all kind of be mixed, and then you have two new mixed varieties from the complementary side and the the new side. But that's not how it works either. The way it works is what you see up here, the semi-conservative, where you have the original DNA, you peel it apart and then put on the new nucleotides on each side, and then essentially you have hybrid DNA, half from the old and half from the new. So this is the way it works, the semi-conservative replication. And again, it took lots of different experiments for them to figure that out. One of the ways they did that was looking with bacteria, where they grew bacteria with nitrogen that had a heavy isotope. Remember how I told you they might use different types of DNA or nucleotides, maybe change the atoms a little bit, make it a little bit heavier. An isotope, if you're not familiar with that, is basically an atom that has an extra neutron. So we know that nitrogen has 15 electrons normally, 15 protons normally, or sorry, I'll take that back. Rewind. 14, nitrogen normally would have 14 Now I'm confusing myself. I have to look it up. But it would have 14 nucleotides. Okay. So normally, nitrogen would have 14 electrons, 14 protons, and 14 neutrons. A radioisotope that's heavy would have an extra neutron. Does that make sense? Did I get you back on track? So it has an extra neutron. So it would still have 14 electrons, in quotes, 14 protons, and 15 neutrons. So it's heavier. It has an extra amount of neutrons, so it's heavier. Does that make sense to everybody? And so that's a radio heavy isotope that was mixed into this medium, and the bacteria grew in it. So the bacteria grew with 15 heavy isotope of nitrogen. And so then they had DNA that would have this heavy extra neutron. And then you had other bacteria growing in the normal nitrogen. So you had bacteria growing in heavy and light, and then they could take out the samples and break the DNA open and extract the DNA from the bacteria and compare it. So they suspended the DNA in this chloride solution, centrifuge it, and then they weighed it. And so they found that the control group, the DNA was up here because it's all 14, remember? And then those are just fed on the heavy radioisotope. All the DNA would float to the bottom because it was heavier. But it spins, right? Heavy stuff is on the outside. But the new DNA was somewhere in the middle. So that meant that the DNA was a mixture of light and heavy, and thus semi-conservative. And then when they mixed it in with other bacteria, they would see that it was floating near the top and in the middle. So this provided evidence that there was a semi-conservative amount, the semi-conservative replication was taking place because half of the radioisotopes and half of the new isotopes would come together. After, as you remember, take the 15, put it into so this would be the control right here. Go straight from DNA that was heavy. And then they would have these going mixed in with the different mediums, only 0 minutes here, so not enough time. More replication took place over here. Are you guys all confused? What I'm hoping you get the gist is that this provided evidence for this semi-conservative method. Otherwise, the nucleotides would be in a different location after spinning. Irrespective of whether you understood what I was trying to explain, what is the take-home message again? Semi-conservative replication is the way DNA replicates. How does it actually work? Well, there's some different enzymes involved. There's helicases and primases and DNA polymerase. How do we know it's an enzyme, by the way? What do we know about enzymes? Well, they all have ACE at the end. So helicase unwinds the DNA. So you got double-stranded DNA and you unwind it, that's helicase. Primases put down primers that help in putting in little snippets of the complementary strand, and you'll see a picture to help you to understand that. And then DNA polymerase adds nucleotides to the complementary strand. The pictures are worth 1,000 words, so let's take a look at some pictures. Here's our template strand, and DNA is replicating. And what happens is new nucleotides come in. So after you've pulled them apart, these new nucleotides come in, and this thymine will attach to this guanine. What happens is, here we see thymine triphosphate. These two phosphates will get kicked out, and then this phosphate will attach to the hydroxyl end. Hydroxyl meaning OH. Again, if these big words might be a little confusing to you, just realize all I want you to get is that the DNA polymerase sticks the nucleotides on. So this would have been all empty, and then DNA polymerase would bring in the guanine, bring in the adenine, bring in the thymine, bring in the guanine, and then there's an A here, bring in the thymine. And it'll stick to this side, and then there'll be hydrogen bonds holding in the middle. Notice that there's another adenine. Well, that means another thymine will be coming in. And who's the enzyme responsible for putting it in? The DNA polymerase. So what is the job of DNA polymerase? It reads that strand and puts in the complementary nucleotides. That's what I mean by that. Again, it's much more complex than that picture, right? There's gonna be lots of different enzymes, maybe how it's how the enzyme holds it, and so forth. But that's what it does. So what do we see in this picture? Well, this is the original DNA. This is the double helix. And what is helicase doing at this moment? It's unwinding the double strand. You see that? It's unwinding. You have two shoelaces together, and then something's unwinding it, pulling it apart. The DNA polymerase on this side is putting in the nucleotides on the naked strands, what they call it. So that means it's going to continue putting the nucleotides in. You see that? That's on the 3' end, by the way. So this is the leading strand. There's also a strand that's kind of what they call the lagging strand, which requires these primers to come in and hook up with the codons of about 3 pairs or so. At least that's what's shown in this picture. And then this enzyme starts putting in the rest of the nucleotides. And that's done with the primase. You see that? So a short piece of RNA is termed the primer, then the primase is this purple thing here. By the way, if you're just looking at circles to you, realize that helicase, again, is an enzyme. It's a huge protein. This is a huge protein, primase. And then these are little segments of primer. Now, why do they call it the lagging strand? Because it's lagging behind this strand, the leading strand. So this one's getting done just a little bit quicker than that one. And then eventually you're going to end up with 2 new strands of DNA. So this is how DNA replication happens in a simple understanding, right? Super simple way. The primers will be removed and replaced with DNA. There's another enzyme that will join the ends of the newly synthesized DNA back together. So as helicase is unwinding the DNA, the leading strand is getting new DNA in, and then the lagging strand is getting its DNA. And then you can see the DNA polymerases are at work. And then ligase comes in and joins these pieces together from the different primers that were found on the lagging side. So there'll be little gaps, and they'll help join it back together by getting rid of the RNA and putting the complementary DNA into that portion. So that's the job of ligases. It's basically fixing the lagging strand. But either way, you end up with 2 new strands of DNA. And if nothing else, is these pictures pretty simple to understand? You can see it's unwinding. DNA polymerase is throwing in new bits. There's little primers down here, and it's more complicated, but DNA polymerase is filling in the gaps after the primase has been in there. Ligase comes in and patches some of the holes on the lagging strand. But again, in a zoology class, introductory class, I don't want you to worry about you being an expert of this. Just to understand the nutshell. Helix unwraps it. DNA polymerase fills in the gaps on one side. The other side, the lagging strand, leading strand. Just kind of remember those kind of terms and the basics of what's going on. Now, what about chromosomes? Where does this all come into play? Well, chromosomes are basically when you have DNA all wrapped up tightly before mitosis, before meiosis, right? Before that, it's just floating around. So it's like you got, here's another silly analogy. It's like your spaghetti. Your spaghetti is in the bowl. The DNA is off, the DNA is the spaghetti. You're ready to eat some, or are you just going to grab one piece? What you do is you're probably going to get your fork, spin it up. That's your chromosome, essentially, but done on a molecular level. An embryologist, as you can imagine, might have saw it first. He at least and described it first, and that would have been Walter Fleming in 1882. He saw chromosomes under a microscope. The number of chromosomes varies enormously from species to species. For instance, there's this Australian ant that only has one pair of chromosomes. There are some ferns that have more than 500 pairs of chromosomes. That's a lot more than you, by the way. You have 23 pairs. So you might be thinking that lowly fern is a lot less complicated than you, but in some regards, molecularly, it isn't. Chromosomes exist in somatic cells. Remember, somatic cells are the cells of your body as pairs. Homologous chromosomes are identical pairs. And then, and so forth. Chromosomes are composed of chromatin, which is a protein DNA complex, where it's about 60% protein and 40% DNA. A typical human chromosome, so we're talking about one chromosome in a human, has 140 million nucleotides. So all those A's, T's, C's, and G's, 140 million just for one of your chromosomes on average. We're obviously not talking about the Y chromosome of guys, but the typical chromosome, because the Y chromosome is a lot shorter, a lot smaller, right? And believe it or not, that chromosome, if it could be stretched out end to end, would be 5 centimeters long. And it would represent about 2,000 printed books of 1,000 pages each. That's incredible. And then, of course, it's all controlled without librarians and coiled up in DNA. So you don't have to check out the book or anything like that. The DNA is wrapped up positively charged proteins called histones. Let me show you some pictures. This will all make sense in a second. 200 nucleotides of DNA coil around a core of 8 histones, forming what they call a nucleosome. And then these nucleosomes coil into solenoids. Solenoids are then organized into looped domains. The loop domains appear to form in rosettas. All that's fancy. Let's take a look at some pictures. I think that'll make it even easier. Maybe. Here's our chromosome. We're going to cut a piece and take a look. We're going to magnify it in. Now you can see the rosettas. They're on some type of scaffolding protein. Let's zoom in some more. You'll see that these rosettas come off with the chromatin loops and some kind of scaffolding protein. And then those DNA gets wrapped around these other proteins that are the histones. So you can see DNA is literally looped around these balls of histones. And together we call that a nucleosome. And then it even has a little protein that clamps it. But we're talking about 8 little pieces for each one of these. And then here's our DNA unraveled, single-stranded. Isn't it unbelievable? All this is happening in each of your nuclei. Pretty amazing. And then every animal, every plant, for the most part. Here's another picture. DNA is all singles, here's our double helix DNA. Then it gets wrapped up in all these histones, and then we call it a nucleosome. And those get wrapped up, and they get wrapped up, and they get wrapped up, and all sorts of different wrappings. And eventually you have a chromosome that you see during mitosis and meiosis. And remember, it's replicated at this point, so you have a sister chromatid held at the centromere at the middle that will get pulled apart during anaphase. Another cool picture, just wrapped around. This keeps getting wrapped around in every possible manner until you have yourself a chromosome. And again, it's held together at the centromere. And then these would be called sister chromatids. And then this is what they look like under a microscope, electron microscope, I believe in this case. This is what we call a karyotype. So during mitosis or whatever, or meiosis, once we can pull these out and we look at our homologous chromosomes. This is homologous to this, right? It's not homologous to that. Those two together are homologous. And you can tell they've been replicated, and there's a centromere in the middle. So here's 23 pairs of chromosomes representing the karyotype of a person. And here's our X and Y, our sex chromosomes. We've already kind of hit on some of this, but here's a chromosome that replicates and then comes together, and we have our homologous chromosomes held together at the centromere. And as I already mentioned before, and you already know, humans have 46 chromosomes or 23 pairs because we're diploid organisms. And we call that a karyotype when it's displayed like this. I appreciate your attention. I hope you have a great weekend, and take care.